Thanks, Ashley, and good morning from LI-COR Biosciences. Today I'm going to talk to everybody about measurement of CO2 evolution in a multiplex Flask system using the LI-8100 soil CO2 flux system. I'd like to start by talking about what we're trying to accomplish with this webinar, why somebody would want to go about making these measurements. Then I'm going to move into how you actually physically make the measurements, optimizing the 8100 system to make the measurements, how you actually configure the 8100. There's some plumbing changes that are made, and there's some repurposing of software fields. And then I have two practical measurement examples to show you where we look at where we measured CO2 fluxes from isolated detached fruit. And then we'll talk to you about ordering information, sort of the parts that you can get from LI-COR that are necessary to make these measurements. What we're doing is we're providing a solution for making repeated CO2 flux measurements from multiple discrete samples. In its normal configuration, the LI-8100 soil CO2 flux system is used to measure CO2 fluxes from soil. So you have a chamber that sets over a continuous body of soil and it accumulates CO2, and we calculate a flux from that rate of accumulation. And this allows you to explore both temporal and spatial variation. So within the system, there's a multiplexer that'll let you connect up to 16 chambers to it at once. And so you can look at temporal variation by making repeated measurements with the system, and you can look at spatial variation by making measurements across a landscape. With this system, with these modifications to the system, samples are placed in sealed chambers. And so you have a discrete sample in a sealed chamber. And rather than looking at temporal and spatial variation, now because you're looking at multiple samples, you can still make repeated fluxes, so you still get the temporal variation component, but instead you get a high throughput system because you can make measurements on up to 16 samples at once. So this leads into the why you'd want to do this. Well, the system's extremely sensitive. It's able to resolve small fluxes from minimal sample material. We took this system out and configured it based on some of the information I'm about to show you in a couple of slides, and we were actually able to detect carbon fluxes from a single fruit fly with it. Because of the multiplexed Flask arrangement, it allows you for high throughput, and this becomes important when you have lots of sample material available at one time. So if you're looking at post-harvest physiology and the particular fruit that you're looking at all ripens at the same time, this gives you high throughput because you can process 16 samples at once. And because of the way we treat samples between measurements, we actually add an auxiliary pump to the 8150, and that serves to flush chambers out with ambient air between measurements. You're able to make repeated measurements on the same sample without actually having to open the chamber or have any involvement. So how do you go about doing this? Well, you read the application note that accompanies this webinar, and you listen to the lecture. The first thing it's going to direct you to do is to define an operating envelope. And what we're talking about here is really selecting an appropriate chamber size for the material that you're going to be working with. And this has to do, again, with physical size of the material, but it also has a lot to do with the actual fluxes that you're going to measure. And I'm going to talk about that here in a second. The next thing you'd have to do is replumb an 8150. You'd have to install an auxiliary pump. You'd have to interface this with the normally open side of the solenoid manifolds. And then when it actually comes time to make the measurement, there are a lot of fields that make sense for making soil flux measurements with the soil flux chambers that either don't make sense when you're measuring discrete samples or their meaning changes significantly. And so we talk about repurposing some of the software fields in the LI-8100 software. And then the final thing I want to talk about in terms of how you actually do it is how you can get an integrated temperature measurement with the flux measurements. Okay, so the first thing you want to do is to select an appropriate Flask size. And the question comes, well, why does Flask volume matter? And Flask volume matters because the way the 8100 system works is that it makes flux measurements using a closed transient system approach. And in this approach, you're measuring CO2 concentration, the change in CO2 concentration over time. And so if you fit a curve to the relationship between CO2 concentration and time, and you take the slope of that curve, that's the term DCDT. And the way you get from DCDT to flux is by multiplying by the ideal gas law. And when you do that, you see that there's really three variables outside of DCDT that are important in getting to the flux. And that's the temperature inside the chamber, the volume of the chamber, and the pressure inside the chamber. Over a range of typical atmospheric pressures, the influence of pressure is pretty minimal. Over a range of biologically significant temperatures, the influence of temperature is pretty insignificant too. A change from 20 to 40 degrees C causes about a 6 to 7% change in the flux for the same DCDT. But if you change the volume, it's almost a one-to-one relationship between the change in volume and the change in flux for a given DCDT. So if you double the volume, you cut DCDT in half. And then DCDT is also important because you can think about it in the same terms that you think about the error due to instrument noise. And so, infrared gas analyzers can be described as having noise, described as having peak-to-peak noise. So the change in apparent CO2 concentration from one moment in time to the next moment in time due to noise in the instrument. And then chamber volume also has an influence in terms of mixing. And mixing is very important. I'll talk about that more here in a second. So let's start with this relationship between DCDT and error in the measurement due to instrument noise. For the LI-8100 system, the peak-to-peak noise of the instrument is well known. And what we've done in the application note is we've actually modeled this noise for various sampling lengths. And we've plotted that against the amount of error that you would expect in the estimation of DCDT for these different sampling lengths. And so what you see here on the x-axis of the figure is DCDT. And on the y-axis is fractional error. So you can think about a fractional error of 0.1 being a 10% error in the flux measurement. And so what we see is that as DCDT gets smaller, the error in the measurement has the potential to get much larger. And we can partially ameliorate this effect by increasing the number of observations that go into the estimation of DCDT. And so with more observations, a longer sampling length, we reduce the effect of error. But it's much more effective at reducing the effect of the error if we can just make measurements with a larger DCDT. And so the idea here is that for any experiment, there's a certain acceptable level of error in the measurements that you're going to make, whether it's 5%, 1%, 10%, it doesn't matter. But you know this going into it when you're setting up the experiment. And so you can look at this and you can say, well, I need to make measurements with this level of precision. And to do that, I need to be measuring fluxes that give me a DCDT larger than whatever value, 0.01. Okay, and so this sounds silly at this point because we're talking about selecting the value of what it is that we want to measure, but it'll all make sense here in a second. So taking our knowledge of where our minimum DCDT needs to be, we need to go out in the literature or make some test measurements and figure out what magnitude of fluxes we're going to be measuring. Because if we know the fluxes, or have a guesstimate of the range of fluxes that we expect to be working with, and we know what the minimum DCDT is that's acceptable for the minimum amount of error or the maximum amount of error we want to see in our measurements, we can select a chamber volume that's appropriate to make those measurements. And so what you see here is the raw flux, so a flux not standardized for area or mass, plotted against DCDT for various chamber volumes. And so you can either take the flux equation presented in the application note, or you can take this figure and you can make an estimate of what sort of chamber volume you need to be working with. The next thing we want to think about when we're talking about selecting an appropriate chamber volume is mixing inside the chamber. And so when you're making these measurements, you're subsampling the chamber headspace with a gas analyzer. And the assumption is that your subsample is representative of the bulk air inside the chamber. And if it's not representative, you can get an error in the measurement. And what we talk about is we want the chamber volume to be well mixed. So we want all the air inside the chamber to be at the same CO2 concentration. Because if it's not, we're either sampling pockets of air that aren't at the same concentration as the bulk air, or we have pockets of air in there that are at different concentrations, either lower or higher CO2 concentration. And diffusion of CO2 in and out of these pockets adds an additional error term into the measurement. Now, mixing has a lot more influences than just chamber volume. It has to do with the geometry of the chamber itself, the kind of sample material that's in there, the flow rate through the chamber, whether there's additional fans in the chamber, and all of these things affect the amount of error that can come from mixing. And so this is nearly impossible for us to model in an application note or to present you with a way to do it. And so what we do is provide a very simple example of how we determine whether or not mixing is good in a chamber. And so the figure that's presented here, flux is on both axes. And on the x-axis, our flux is measured with chamber fans turned off. So what we have is a 4-liter chamber connected to an infrared gas analyzer, and there's a pump that circulates air between that chamber and the infrared gas analyzer. And inside the chamber are some fans. And so each point on this graph represents a single sample with 2 flux measurements made on it. One was made with those the fans in the chamber turned off, and one was made with the fans in the chamber turned on. And in a situation where we have good mixing inside the chamber, due simply to the pump that circulates air between the chamber and the infrared gas analyzer in this case, we would expect a 1-to-1 relationship between those measurements. And that's exactly what we see. In this case, the slope is nearly 1, the intercept is nearly 0, and we have a good correlation between the data, indicating that in this example there's sufficient mixing inside the chamber. So once we've selected a chamber volume that we need to work with for our material, it's time to go and attach those chambers to the system and modify the LI-8150, the multiplexer, to actually make these measurements. And so the first thing you're going to do is you're going to install some plumbing fittings that give you access to the normally open side of the solenoid manifolds. In its factory configuration, when the system leaves LI-COR, these sides in the manifolds are sealed because they're not actually used for anything. The next thing you're going to do is add an auxiliary pump inside the 8150, and what this pump does is it serves to flush the chambers with ambient or conditioned air between measurements. And what this does is it keeps the chambers at near ambient conditions when you're not making measurements. This is important because otherwise, if we just leave the chamber sealed, CO2 is going to continue to accumulate in there beyond the measurement period, and eventually the chamber, the airspace inside the chamber is going to come into equilibrium with the airspace inside the sample. And since what we're essentially measuring are diffusive fluxes, once there's no gradient there, there's no more flux. And in terms where you wanted to look at something where the CO2 concentration or water vapor concentration or some other trace gas inside the chamber influences the flux rated, it's really important there to keep it at near ambient because you're preventing these biological interactions between the gases in the chamber and the sample itself. But the nice thing about this system is that with the pump unpowered, the 8150 operates as normal. The only difference is that the mounting hole we use to mount the auxiliary pump is the same hole that's used to mount a splash guard inside the instrument, and that splash guard has to be removed to install the pump. So how does this all work? Well, inside the LI-8150 are banks of solenoid manifolds. There's 2 shown here in this flow diagram. There's a flow meter, there's a pump, and there's a subsampling loop. So everything shown here besides the chambers and the LI-8100 analyzer control unit are housed inside the 8150. And what we're showing here is a schematic for the way that it's normally configured to make measurements. So in this case, the solenoid at port 3 is active, and it's circulating gas between the primary circulation pump in the LI-8150 and the chamber, and the LI-8100 is pulling off of this primary circulation loop in a subsampling loop to actually make the measurement. When we configure the system for Flask measurements, we add an auxiliary pump to the normally open side of the solenoid manifolds. And so the measurement loop doesn't change. It's the same thing shown with blue arrows that was shown in the previous slide. But what does change is now that all the flasks that aren't being measured have ambient air being pushed through them. The effect of pushing ambient air through the chambers goes like this. What's shown here are CO2 concentrations inside the chambers for a bunch of respiration measurements made on bananas. So these are hourly respiration measurements made over a 3-minute time period for each chamber. And in this system, there are 6 chambers being measured. There's 6 chambers connected to the system at once, and there's only one being measured at a time. So if you're moving from left to right along the x-axis, where you get to the first up arrow is where the first measurement's being made on the first chamber. And so the system switches from flushing the chamber out to being a sealed volume, and CO2 is accumulating, so the CO2 concentration increases. When you reach that first down arrow, that's where the measurement stops, and we begin flushing the chamber out with ambient air again, and you see the CO2 concentration in the chamber rapidly drops down to near ambient levels. As we keep moving left, we get to this little point where it bumps up, indicated with the red arrow. And what that is so that's the point where the system switches from measuring one chamber and flushing 5 chambers to just flushing 6 chambers out, and so the flow rate drops. And when you're flushing a chamber like this with ambient air, it's essentially operating as an open system. And so we're in the closed system, the CO2 concentration in the chamber is dependent on the chamber volume and the flux releasing CO2 into the chamber. In an open system, the CO2 concentration in the chamber is dependent on the incoming CO2 concentration, the flux, and the flow rate through the system. And so in this case, the CO2 concentration in the chamber increases slightly because the flow rate through the chamber decreases. In terms of software configuration, most of the stuff that needs to be changed occurs under the port setup tab in the software configuration utility. For the chamber, you're going to select custom, and the chamber volume is going to be the volume of your chamber plus any tubing that's used to connect it to the 8150. There's a box that says this chamber provides a closed signal, and you're going to have this unchecked because the chamber doesn't provide a closed signal. But soil area is really the first space where the meaning of the field really changes. And so it could mean the same thing. You could treat this as surface area of the sample and enter it in centimeters squared. Or, for a lot of applications, you're going to want a flux on a mass basis. And so if you were to enter the mass of the sample here in decigrams, you get a flux in micromoles per kilogram per second. Whereas if you enter it in centimeters squared, the surface area of the sample, you get a flux in micromoles per meter squared per second. The chamber offset becomes meaningless in this situation. So in its normal application, the chamber offset's used to correct for the volume either added or displaced by the soil collar. But in this case, we're dealing with a sealed chamber, and so the chamber offset becomes meaningless. And so this is always going to be 0 for your measurements. The extension tube volume changes fairly significantly in terms of its meaning. So we've said multiple times that volume is the single most important factor in the relationship between DCDT and flux for a closed system. And so if you have a chamber, and you've entered a chamber volume, and then you place a sample in there, and that sample occupies some significant portion of the chamber volume, there's going to be an error in your volume term unless you account for the volume of the sample. And so you can do that by entering the volume of the sample for the extension tube volume, but you enter it as a negative number. In terms of a temperature measurement, we have soil temperature thermistors and a chamber sensor interface box that lets you connect up to 3 of these sensors per port. And this provides a very convenient way of getting an integrated temperature measurement. The only place that these would not be appropriate is where you're looking at rapid fluctuations in temperature. So these are thermistor-based temperature sensors, so they have a relatively significant thermal mass to them. And so if you have rapid oscillations in temperature, you're not going to detect that with these sensors. So in terms of measurement examples, in preparing the application note, we wanted to verify that this actually works the way that we thought it was going to work. And so we went out and we measured respiration from detached fruit. In our first set of experiments, we looked at respiration from 12 navel oranges over a 24-hour time period under ambient conditions. And in the second set of experiments, we looked at banana respiration and made hourly respiration measurements for 1 week as the bananas ripened from stage 2 to stage 7. So anytime that you're looking at flux data, we like to talk about quality controlling the data. And one of the things that you want to do that is you want to look at the actual raw plots of CO2 concentration over time. And what we have here on the top is the CO2 concentration is in an empty chamber made simultaneously with the orange respiration measurements. And on the bottom is the CO2 concentration in a chamber containing an orange over the 3-minute time period that we made the measurements. And the first thing to see is that there's really a sizable carryover of CO2 in the measurement loop between measurements. And what this represents is, if you think back to the schematic that I just showed you, there's 2 volumes here that are discrete from each other. There's the volume of the chamber and the tubing used to connect it to the multiplexer up to the solenoid ports. And then on the other side of the solenoid manifolds, there's all the tubing and pump volumes and whatnot between the solenoid manifolds and the LI-8100. And so when you switch from chamber to chamber, there's a volume of gas trapped between the solenoid manifolds and the 8100s that's going to be at the maximum CO2 concentration from the end of the measurement at the previous chamber. And when you switch to the new chamber, it takes some time period for that to flush out and come into equilibrium with the chamber before you can start detecting the increase in CO2 concentration in the chamber that you need to make the flux measurement. And so what we do here is we just simply apply a 60-second deadband at the beginning of the measurement. So if we ignore the first 60 seconds of the measurement when the whole system's coming into equilibrium at the beginning of sampling a particular chamber, we get a relatively good fit. And that's what I'm showing here with the black lines. And so those lines are fit to the data from 60 seconds on. So what do the fluxes look like? Well, the fluxes look really good. They are comparable to what we measured in preliminary work with an LI-6200. They all sort of track each other, and they all show this interesting decrease in flux over the time period of the measurement. As I said before, these measurements were made under ambient conditions, so we didn't control for chamber temperature. And if we plot the flux against chamber temperature, we see that they correlate well with each other, and that the change in temperature accounts for about 79% of the change in flux over the measurement period. In our banana example, what this actually illustrates is the ability of the system to really make repeated fluxes for long time periods. And so what we had here are 6 chambers, each containing a banana in a temperature buffered environment. So we had a water bath inside of a large cooler, and the chambers were inside that. And we made hourly flux measurements for a 1-week period on the same bananas as they ripened from being very green, stage 2, to very ripe, stage 7 bananas. In terms of what you actually need to make the measurements with the system, well, you need an LI-8100 that has the infrared gas analyzer, the computer that controls everything, and the data storage. You need an LI-8150 multiplexer. That's what allows you to connect more than one chamber to the 8100. And we offer these in both 8 and 16 port options. In the application note, we recommend for powering the system the 8150-770. This is an AC power supply, and it provides 4.5 amps of power. We offer the parts kits that include the pump, all the fittings needed to modify the 8150, and then all the fittings and tubing that you would need to connect either 16 or 8 chambers, depending on which multiplexer you had, to the LI-8150. If you wanted the integrated soil temperature integrated temperature measurements, we have the soil temperature thermistors and chamber sensor interface boxes that can be purchased separately. And so really the only thing that we don't provide to make these measurements are the flasks themselves. Well, that's the end of the presentation, and so I see that we have some questions from the audience, and we'll move on to the question and answer section now.
All right, it looks like we're ready to go here. Our first question is, are there limitations to how small or large a chamber can be?
Yes, there are probably limitations to chamber size. On the small end, as the chamber volume approaches that of the tubing and analyzer volume inside the LI-8150 and LI-8100, any small errors that result from estimating the system volume become more important as they represent a larger fraction of the total volume. The volume inside things like fittings and pumps and the plumbing, it's often difficult to estimate the exact volume inside there, and often things like fittings and pump volumes get ignored. Because with typical system volumes where you're talking about a total volume that's on the order of liters, these things account for fractions of a percent of the total volume. On cases where you want to use small chambers and the system volume is, fractions of a liter or less, they can account for a much larger percentage of the total volume. On the large end, the maximum size will be contingent on the fluxes from the sample material and the ability to provide adequate mixing inside the chamber. If you remember, as the chamber volume increases for a given flux, DCDT becomes diluted at the same rate. So if you double the system volume for the same flux, you cut DCDT in half. So under these conditions, it's quite possible to have a chamber that's too big to measure your sample material.
Thanks, Jason. Our next participant asks, how small of rates can be measured with this system?
Well, with a properly configured system, the limits to rates on the small end are really small. In tests that we've done here, we've been able to detect CO2 being involved from a single fruit fly in a chamber. However, because these kind of measurements require the use of very small chambers, there are a lot of subtleties to the measurement that aren't well covered in the application note or in this webinar. And so if you're interested in making these measurements, it would be best to contact one of our applications people first.
Thanks, Jason. Our next question is, how frequently can individual chambers be sampled?
The frequency with which you can sample a chamber is dependent on the number of chambers you're using and the observation length used of each chamber. Because they're sampled sequentially, you can think about it being additive. So, for example, if you had 10 chambers and you were using an observation length of 3 minutes, you could sample each chamber every 30 minutes.
All right, thanks, Jason. So how important is measuring the temperature in the chamber?
Well, as I said earlier in the presentation, the effective temperature on the measurement's fairly small. For the same DCDT, changing temperature 20 degrees Celsius only changes the flux by 6 or 7%. If you don't measure chamber air temperature using this system, the chamber temperature defaults to the temperature of a reference junction inside the instrument. So if the temperature differential between your chamber and the instrument is small, the effect on the flux is going to be relatively small. However, if you're expecting large temperature differentials, like you want to make measurements far away from ambient or whatever conditions the instrument's going to be housed under, it becomes much more important to measure temperature in these conditions. And the thing to do there is just to measure it and recompute the fluxes after the fact using the correct temperature.
All right, great, Jason. It's looking like that's going to have to be our last live question. I want to thank everyone for submitting your questions, and if you haven't yet got an answer, just to stick around and a panelist will get back to you as soon as they can.